Okay, so we're going to return to talking about mRNA decay. And today I'm going to focus on two pathways that are mechanistically related but serve distinct purposes. And again, I will be focusing on mammalian cells. The first one is nonsense-mediated mRNA decay, which we abbreviate NMD. And the second is Stauffen-1-mediated mRNA decay, which we abbreviate SMD. And today I'm going to be focusing on work that's done in my lab. We'll start with NMD. And the bottom line is NMD provides for a quality control mechanism to ensure the quality of gene expression by monitoring mRNA translation. And in particular, NMD degrades mRNAs that prematurely terminate translation. And you'll see why this is important. So I'd just like to review. I think you all know from Harry's talk and before that translation normally terminates at the 3 termination codons that are shown here in red. These codons usually don't encode any amino acid, and they signal the release of the nascent peptide and also dissociation of the 2 ribosomal subunits. I started studying NMD, I won't tell you how many decades ago, but our studies uncovered 2 important things. One was that a number of human diseases are due to frameshift or nonsense mutations that result in the premature termination of translation. And the disease is due to the lack of full-length functional protein. We also found that when translation terminates sufficiently prematurely, then there will be NMD. And this is important because even though the disease is due to the lack of full-length functional protein, the presence of truncated proteins from mRNAs that prematurely terminate translation but escape NMD is bad for cells because the truncated proteins can be toxic to cells. And for those of you who want a quick review about nonsense mutations and frameshift mutations, nonsense mutations are usually single nucleotide changes that convert a coding codon to one of the 3 nonsense codons. Frameshift mutations occur in the open translational reading frame, as do nonsense mutations, and they consist of an insertion or a deletion that is not a multiple of 3, shifts the reading frame, and generally generates a premature termination codon downstream of the insertion or deletion. So I described yesterday how many steps of post-transcriptional gene control are mechanistically related, and you've heard from many other speakers examples of this. And it turns out our studies of NMD have provided links as well. We uncovered a remarkable link between pre-mRNA splicing in the nucleus and mRNA translation and degradation in the cytoplasm that's provided by exon junction complexes, which are complexes of proteins deposited upstream of the exon-exon junctions of newly synthesized mRNAs, and they stably contain some of the UPF NMD factors. You've already heard from John about UPF1. UPF1 transiently associates with the EJC. UPF2 and UPF3 or 3X are stable constituents of the EJC. And you'll see more about that in a minute. And we also, and I should say the EJCs were discovered largely by Hervé Leyer, and this was a collaboration with Melissa Moore's lab, Melissa Moore in particular. And Yasu Shigaki, when he was a postdoc in the lab, uncovered a new template for protein synthesis that we call the Pioneer Translation Initiation Complex. It turns out that NMD is a consequence of nonsense codon recognition of premature termination codon PTC recognition during a pioneer round of translation. This is the first round of translation. It utilizes newly synthesized mRNA in the cytoplasm that has maintained the 2 cap-binding proteins that are acquired by pre-mRNA in the nucleus. And if the mRNA underwent splicing, then the Pioneer Translation Initiation Complex will also contain the post-splicing EJCs. We were very surprised that this mRNP could be translated. Fabrice Lejeune showed that it is a precursor to this mRNP, which produces the bulk of cellular proteins. Most of cellular translation uses this mRNP because it's the most abundant. But you can see why NMD would target this mRNP and not this one. For one, CPP80-20 bound mRNA is associated with the EJCs and the UPF NMD factors, whereas by the time CPP80 and 20 is replaced by the major cytoplasmic cap binding protein called EIF4E, it's an initiation factor just like CPP80 and 20, we don't detect the EJCs and UPF proteins anymore. And it gets more interesting than this, and I will show you later that CPP80 actually promotes NMD. It's not just passively there, it activates NMD. So how did we figure this out? We performed immunoprecipitations of cellular RNP. And as an example, transiently introduced into cultured mammalian cells a test plasmid that was either PTC-free, which we call norm, or contained a PTC that triggered NMD, and we call that TER for termination. We also co-introduced a reference plasmid that controlled for variations in transfection efficiencies and RNA recovery so we could have a quantitative analysis. Generated total or nuclear and cytoplasmic fractions under conditions that preserve RNP, because what we want to look at is RNA and its associated proteins. The fractions were cleared by incubation with protein A agarose beads, and then the immunoprecipitation was performed with antibodies bound to the beads. So alpha stands for antibody, anti-CPP80, anti-EIF4E. These are the two cap binding proteins of newly synthesized and steady-state mRNA. They are both rabbit antibodies, so a control for nonspecific IP could be, and in our case was, normal rabbit serum. Then protein was purified for Western blotting, and RNA was purified for RT-PCR analysis. And the next slide shows a Western blot. This was done by Fabrice Lejeune when he was in the lab. He analyzed nuclear and cytoplasmic extracts. The immunoprecipitation was done with these antibodies. And if you look at the resulting Western blot, in the case of the anti-CPP80 IP, he obtained CPP80 and not EIF4E. Either one or the other cap binding protein is associated with a particular mRNA. And the EJC constituents were detected in the anti-CPP80 IP. They were not detected in the anti-EIF4E IP. And that's true for either nuclear or cytoplasmic fractions. So this was the first indication that the EJCs go out with newly synthesized CPP80-bound mRNA into the cytoplasm where translation occurs. The next is an RT-PCR analysis of the test transcript, which was globin, either PTC-free norm or PTC-containing TER, and our reference transcript, MUP, which is not expressed in the cells we are using. And before IP, you can see that the level of PTC-containing mRNA is 20% of normal. After a CPP80 IP, 17% of normal, and EIF4E IP, 12% of normal. And these are very early IPs. We've really gotten this down to a quantitative analysis now, but these numbers are in essence the same. And because CPP80-bound mRNP is a precursor to EIF4E-bound mRNP, we can conclude that NMD targets newly synthesized CPP80-20-bound mRNP and not 4E-bound mRNP. So are you following me so far? All right. Even though we uncovered NMD by studying human diseases, NMD functions largely to downregulate abnormal transcripts that are a consequence of mistakes that the cell makes all the time. And this is because truncated proteins that are produced from PTC-containing mRNAs can function in dominant negative or other ways that are toxic to the cell. Therefore, NMD can be viewed as a type of quality control pathway People often call it a surveillance mechanism. And when you think about it, it makes sense for cells to survey their mRNA very early in its biogenesis to eliminate early on the potential and the production of truncated proteins. And this is what happens in mammalian cells. Here is one of many examples of cell toxicity that occurs when NMD fails. And the example I'm giving is premature termination codon, a PTC, in the last exon of the human beta globin gene. So even though this is premature, it does not trigger NMD. The resulting PTC-containing mRNA is stable, but it produces a truncated protein that behaves very differently than the full-length beta globin protein. The full-length beta globin protein can form a tetramer with alpha globin, and this forms hemoglobin and carries oxygen in the blood. But the truncated beta globin protein can only dimerize with alpha globin, and this results in ineffective erythropoiesis and a dominantly inherited form of what is usually a recessive disease. We were really interested in understanding how cells differentiate a termination codon that is normal from one that triggers NMD. And so we established way back in the mid to late 1990s the 50 to 55 nucleotide rule, which says that termination codons existing more than 50 to 55 nucleotides upstream of the last exon-exon junction generally trigger NMD, whereas those closer to the last junction or downstream do not. So you can automatically see why PTCs in the last exon of the beta globin gene don't trigger NMD. We don't think there's anything special about this exon-exon junction, but if one wants to determine the boundary within a particular mRNA where PTCs upstream of it will trigger NMD and downstream of it will not, you look right here. As with every rule, there are exceptions. So for example, if there were a PTC here and a translation reinitiation codon here between the PTC and the exon-exon junction, then NMD will be inhibited. And also, selenoprotein mRNAs contain UGA codons that are specialized in the sense that they reside in cis with a selenocysteine insertion element in the 3' UTR. And this element promotes the incorporation of the 21st amino acid, selenocysteine, at the UGA codon, some of the time, not all of the time. So these are natural NMD targets, and the efficiency with which they undergo NMD directly reflects the cellular abundance of selenium. So we actually did the rat feeding experiments to show that. All right, now that we know the rule, let's go back to this idea that NMD protects cells from the potentially deleterious effects of PTC-containing mRNAs. As I've said, disease results from NMD-induced protein deficiency. There's only truncated protein produced. But the disease may be milder than and different from that caused if the expressed truncated protein were present. So NMD protects many heterozygous carriers of genes with PTCs from manifesting disease phenotypes that would result if the truncated protein were expressed. And I've already talked about the beta globin gene and thalassemia intermedia. So if the PTC resides upstream of the boundary, then NMD occurs and the disease is recessively inherited. But all you need is one allele from either parent with a PTC that's 3' to the boundary, and you have a dominantly inherited form of this anemia. And this is just a partial list of the many diseases by which PTCs downstream of the boundary produce a dominantly inherited disease. We think the most often produced NMD target is alternatively spliced mRNA. NMD downregulates about 1/3 of all alternatively spliced mRNAs in humans, which is remarkable when you think that 90 to 95% of human genes encode pre-mRNAs that undergo alternative splicing. And I've shown here one of many possible scenarios by which alternative splicing can lead to NMD. In this example, the alternative exon is red, and when it's removed by splicing, translation initiates and terminates normally, there's no NMD. But if it were retained and it had a termination codon in frame with the initiation codon, then there will be NMD. If it consisted of a multiple of nucleotides that was not divisible by 3, there would be a shift in the reading frame and most likely the generation of a termination codon downstream of the shift. And again, NMD. But we think from work that we did with Ben Blenko back in 2006 that most of alternatively spliced mRNAs that are targeted for NMD are junk. They're mistakes. And we think that for a number of reasons, and I've listed 2 here, one of which they're produced at such low cellular abundance that they are not likely to contribute to cell physiology. And secondly, there's rarely tissue-specific regulation, which is a hallmark. Tissue-specific regulation is a hallmark of productive alternative splicing. That said, NMD is used by cells for homeostatic purposes. And an example is provided by many RNA binding proteins. The one I want to talk about here is polypyrimidine tract binding protein, or PTB. It actually feeds back on the splicing of its own pre-mRNA. So normally, PTB is produced with exon 11 retained within the pre-mRNA. But when the intracellular concentration of PTB becomes too high, PTB feeds back on its own splicing pattern so that exon 11 is skipped. This results in a shift in the reading frame, the generation of PTC in exon 12, and NMD. All right, I want to review mechanism quickly. And before I do so, I have to explain UPF1, which you've heard a little bit about already from John. It's one of the core NMD factors along with UPF2 and UPF3 or 3X. And these 2 UPFs, 3 and 3X, derive from different genes. We call them 3 and 3X because we mapped this gene when we first described it. Guillaume Seron was a postdoc in the lab. He mapped the gene to the X chromosome. But many other people in the literature call them A and B. They mean exactly the same thing. UPF1 is primarily hypophosphorylated in cells. It's the last of the UPF factors to join an exon junction complex. It's the only one that's not a stable constituent of the EJC. And it's thought to join when translation terminates sufficiently upstream of the EJC. And at that point, it becomes hyperphosphorylated. I also want to describe the Serf Complex, which was discovered by Shige Ono's group in Yokohama, Japan. There's UPF1, and that's the U in Serf. The complex also consists of the kinase for UPF1 called SMUG1, and the 2 translation termination factors that you've heard a lot about from Harry. The E stands for eukaryotic. So because I'm talking about mammalian cells, they're called ERF1 and ERF3. So that is a Serf complex. So here's the model. During a pioneer round of translation, if a PTC exists sufficiently upstream of an exon junction complex so that the terminating ribosome doesn't remove the EJC, that's the basis of the 50 to 55 nucleotide rule. Notice the distance of the EJC, it's about 20 to 24 nucleotides upstream of the exon-exon junction. And as John said, it's that distance no matter what the sequence of the exon is. So if there's a termination that doesn't remove the EJC, then the Serf complex will form at the PTC. And subsequently, UPF1 and its kinase SMUG1 move to the EJC. UPF1 interacts directly with UPF2, and actually SMUG1 interacts with UPF2 as well. Then UPF1 becomes phosphorylated, and that's the way it's activated. Subsequently, there's a step of translational repression that I'll describe in a minute, and then there's mRNA decay. So how did we figure this out? And in particular, what is the importance of CBP80 to NMD? And this is work that was published last year by a postdoc in the lab, Jungwook Hwang. Jungwook followed up on a finding that Nao Hosoda was, when a postdoc in 2005, demonstrated to be a transient or weak interaction of UPF1 with CBP80. And Nao had shown that this interaction promotes the interaction of UPF1 with UPF2. And we proposed at the time, oh, that's probably UPF1 binding to the EJC. And Jungwook actually showed that that is one of the two steps that this interaction enhances during the process of NMD. Jungwook showed that CBP80 interacting with UPF1 promotes the joining of UPF1 and its kinase to the release factors to form Serf. We can see CBP80 actually physically chaperoning UPF1 and SMUG1 to the two termination factors to form Serf. And we are drawing this UPF1 and that UPF1 molecule to be the same. We don't actually know that, but that's probably the most likely scenario. Then CBP80 takes the two that it promoted to form Serf and allows them to join the EJC. This results in UPF1 phosphorylation, translational repression, and mRNA decay. And very briefly, the way that Jungwook showed this was to, for example, map the region of UPF1 that interacts with CBP80 and then express that little region as a piece that inhibits the interaction and none of the other interactions that UPF1 interacts has directly. And was able to show that when this inhibition is blocked, the SMUG1 and UPF1 don't join the termination factors. And when it's blocked, SMUG1 and UPF1 don't join the EJC. The EJC forms, but that last step doesn't occur. He also did the same by using an inhibitory piece that came from CBP80. And then what about translational repression? Actually, I was very surprised by this finding. This was done by Olaf when he was a postdoc in the lab. And Olaf showed that UPF1 phosphorylation triggers translational repression, which generally precedes mRNA decay. That's what I think, no matter what the mechanism. All right, oops. So you already know that when UPF1 with its kinase joins the EJC, UPF1 becomes phosphorylated. And what Olaf showed is that phospho-UPF1 interacts with the two highest molecular weight subunits of EIF3. That is part of the 43S translation initiation complex poised at the AUG initiation codon, waiting for that 60S ribosomal subunit to come on to make a functional 80S complex and begin initiating. But that doesn't happen. So UPF1 binding here inhibits 60S ribosomal subunit joining and translation initiation, blocks the 80S complex formation. And phospho-UPF1 also promotes decay from both the 5' and 3' ends. So we've already talked about decapping and XRN-mediated 5' to 3' decay, as well as deadenylating and exosome-mediated decay. Those steps are inhibited by phospho-UPF1, are promoted, excuse me, by phospho-UPF1. And I don't want to run out of time, but I'll just explain. This slide got a little weird anyway. But let me just explain one of the things that convinced me this was true. Olaf showed that a PTC containing mRNA that initiates translation from the cricket paralysis virus internal ribosome entry site, which doesn't utilize EIF3 in its initiation process, is immune to NMD. Whereas the same PTC containing mRNA that initiates from a encephalomyocarditis virus iris, which does use EIF3, supports NMD. And he chose the EMCV iris because we knew we could debilitate it to a weak enough activity to recapitulate the weak activity of the cricket paralysis virus iris, because we couldn't have any differences in the initiation efficiency. Otherwise, we'd have differences in the termination efficiency. So that's one of the many ways he showed this mechanism. And then yesterday I told you that SMUG6, like The RRP44 constituent of the exosome has a pin domain and an endonuclease activity. So besides the two activities, or the activities that degrade the NMD target from both the 5' and 3' end, there is an endonucleolytic activity accomplished by SMUG6. And quite frankly, nobody yet knows what's the relative contribution of the two pathways to the NMD process. So that's something that people are working on now. All right, so I'm going to shift to SMD. And I want to talk about how it provides for the conditional regulation of specific genes whose mRNAs bind the double-stranded RNA binding protein Stauffen-1 in their 3' UTR. I want to describe how SMD and NMD are competitive pathways, and then I want to talk about how long non-coding RNAs can transactivate SMD. This is a comparison of SMD and NMD. Stauffen, before we began this work, was best characterized in Drosophila as being involved in the export and positioning of mRNA properly in Drosophila oocytes. It also regulates the translation of particular oocyte mRNAs. And we got onto this because Yoon-Ki Kim, this guy right here, who was a postdoc in the lab at the time, used UPF1 as a bait in the yeast 2 hybrid analysis and came up with Stauffen. So to just summarize the pathway, Stauffen binding to an mRNA 3' UTR functions analogously to how an EJC functions during NMD. So when translation terminates, usually normally in SMD, at a point where it doesn't, the terminating ribosome doesn't physically remove bound Stauffen, then mRNA decay is triggered because UPF1 joins Stauffen, and that triggers decay. And that's very similar to NMD, where when translation terminates prematurely, then UPF1 joins the EJC, and that triggers mRNA decay. One big difference is, of course, this does not depend upon UPF2 or UPF3 or 3X. It doesn't depend upon the EJC at all. This will target an mRNA that derives from a gene that has no introns. And also, while NMD, I've told you, is restricted to newly synthesized mRNA because CPP80 promotes NMD, this pathway targets both newly synthesized and steady-state mRNA, and that's what you would expect if it's important for cells to, at some point, downregulate an SMD target, because most of cellular mRNA is bound by EIF or E at the cap. All right, when we have the tethering experiments and show that Stauffen and UPF1 interact directly in mammalian cells and in vitro, I called Luc Farique, Luc de Grossier, who was graduate student Luc Farique, had shown that there were something like 23 mammalian cell mRNAs that they knew were binding Stauffen. And so sure enough, when Luc mapped the Stauffen binding site in ARF1 mRNA, it was binding to the 3' UTR. And what Yoon-Ki did was a phosphoinduction experiment, the way that we talked about yesterday. He took ARF1 cDNA, because you don't need introns for SMD, and took this Stauffen binding site and the sequences that were immediately upstream in the 3' UTR in case the terminating ribosome would remove Stauffen if we deleted it, and drove expression using the FOS promoter, which, remember, is transiently inducible when serum is added to serum-deprived cells. He did this in L cells, which synchronize very nicely in G0 when they're serum-deprived, that had been treated with control or Stauffen mouse. These are mouse L cells. Mouse Stauffen siRNA. There's the Western showing Stauffen was downregulated. And then looked at the level of test mRNA normalized to reference mRNA, which is insensitive to serum levels, as a function of minutes after serum addition to serum-deprived cells. And you can see, as I said yesterday, the burst in transcription is fast and pretty much over by about 30 minutes. So at that point, since there's no ongoing transcription to confound the decay analysis, the half-life analysis, one can look at half-life And when Stauffen is downregulated, sure enough, the half-life of the mRNA is decreased. Whereas in the presence of control, siRNA, SMD is fully functional. And we can conclude that ARF1 mRNA is an SMD target, and that using UPF1 siRNA inhibits decay, just like Stauffen siRNA does, but UPF2 siRNA has no effect, UPF3 siRNA has no effect. Once we realized that Stauffen binding to an mRNA destabilized the mRNA, we realized the best way to identify SMD targets is not to see which ones were binding Stauffen, but to see which mRNAs are upregulated when Stauffen was downregulated using microarray analyses. And we found that about 1% of HeLa cell transcripts were upregulated at least twofold. Not all of these are necessarily direct SMD targets, but we know that a number of them are. And oh, I guess I'm not telling you what they are, but that's okay. You don't really need to know. A lot of them are growth factors, like c-Jun, for example, and we'll talk about serpine-1, which is involved in cell motility. And then the last thing that Yuki did before he went and started his own lab at Korea University was to show that the efficiency of SMD goes up during myogenesis. And this is a tissue culture model for myogenesis where mouse C2, C12 myoblasts, which have a single nucleus, can be fused to form myotubes, multinucleated myotubes. And during that process, the efficiency of SMD goes up. So enter Gong, who is still a graduate student in the lab. And he showed that SMD and NMD are competitive pathways in a way that matters to myogenesis. Bottom line is, UPF1 is in common to both NMD and SMD pathways. It can bind through its CH domain either UPF2 during NMD or Stauffen1 during SMD, which makes the two pathways competitive. And competition explains how the efficiency of SMD is upregulated during myogenesis. So this is a lesson for the students. What Gong showed was that during myogenesis, the cellular abundance of Stau2 decreases. So a priori, you would say, oh, the efficiency of SMD must go down during myogenesis. But it doesn't. It goes up. And the reason why it goes up is because its competing molecule in the NMD pathway, UPF2, decreases more. So all I can say is when you want to draw a conclusion, be sure you've asked the question that gave you the answer you think you have in more than one way, so that you are not being myopic in your interpretation because you don't have the full story. So the competition matters because PAX3 mRNA is an SMD target. And it's important for cells to degrade that during the myogenic process because it maintains, PAX3 protein maintains myoblasts in an undifferentiated state. So to go from myoblasts to myotubes, PAX3 mRNA levels are decreased by SMD. And remember how I told you yesterday, oftentimes mRNA decay is coordinately regulated with transcription, and that is true here. So what Gong did was to use myoblasts and use different siRNAs and always controlled the level of PAX3 mRNA by normalizing it to the level of PAX3 pre-mRNA, which will control for any changes in transcription. And you can see that Stauffen and UPF1 siRNAs upregulate the abundance of PAX3 mRNA relative to its pre-mRNA. And because of competition, UPF2 siRNA, which wipes out NMD, allows the efficiency of SMD to be increased, and therefore the level of PAX3 mRNA to be decreased by SMD. And as we would expect of an SMD target, PAX3 mRNA co-IPs with Stauffen, just like serpine-1 does. We knew that was an SMD target. GAP DH mRNA is not an SMD target. It doesn't bind Stauffen-1 above background. And then in comparing myoblasts to myotubes and the level of PAX3 mRNA relative to PAX3 pre-mRNA, using GAP-DH in each case, while the level of pre-mRNA goes down during myogenesis, the level of mRNA goes down even more, so we can't even detect it. So yes, PAX3 gene transcription is downregulated during myogenesis, and PAX3 mRNA decay is augmented coordinate transcription and mRNA decay. And then myogenin is a natural NMD target. It has upstream open reading frames, which trigger NMD when they're used, and translation terminates at the end of one of them. And it is important to keep myogenin levels up because myogenin is important for the transition from myoblast to myotubes. So it makes sense that this would be an NMD target. And now I want to end by telling you the last story I want to tell you that was just published about a month ago in Nature, where long non-coding RNAs can transactivate SMD. And this is again done by Gong. Remember ARF1 mRNA, it's a SMD target. And remember how I told you that Luc Ferrique and Luc Desgrossier's lab had mapped the Stauffen binding site to the 3' UTR. Well, it turns out, you don't need to go through all of this, I'll just tell you that there's a 19 base pair stem that binds Stauffen. That makes sense, it's a double-stranded RNA binding protein, it's a perfect stem. This 100 nucleotide apex seems to contribute to some of the binding for reasons that we don't know, because we don't really understand what makes a Stauffen binding site. Nobody does, even though it's been studied for a long time in any organism. But we do know that base pairing matters more than the composition of the bases that make up the base paired region. Anyway, we could never find any comparable structure in any other SMD target. So Gong was looking at things, and he found that ALU elements were enriched in the 3' untranslated region of SMD targets. There were 13% of SMD targets have a single 3' UTR ALU element. And when he looked at the human genome and the cDNAs from GenBank, he found that only 4% of those mRNAs had one or more 3' UTR. We focused on single ALU elements and 3' UTRs because we didn't want to be concerned with editing. You know, double-stranded RNA structures can be edited, A to I edited in the nucleus, and that causes the retention. So we're focusing on single 3' UTR ALU elements. So remember, if something is conserved, it probably has a function. And this is a conservation among SMD targets. What are ALU elements? They're short, interspersed elements or signs. They're about 300 base pairs. There are about 1.3 million copies in the human genome. They're commonly found in introns and intergenic genomic regions. They're known to, because they're AU-rich, create polydenylation sites, create splice sites. They regulate gene expression at multiple levels, but they had yet to be shown to have function in mRNA decay. When Gong looked at Serpine-1 mRNA, or FLJ, which has an unknown function, this is involved in wound healing, it's protein, he found that indeed this is what the ALU element looks like, it's in red, and he paired those two because they looked to be able to base pair with this long non-coding RNA. He found there were about 230 long non-coding RNAs that contain an ALU element. This is one of them. And the delta Gs are pretty respectable. Notice that there are at most 2 nucleotide mismatches together before there's base paired regions again. So the hypothesis was this double-stranded structure that was forming between 2 molecules rather than within a single molecule was recruiting Stauffen and triggering SMD. So the first thing he had to do was to take the long non-coding RNA and clone its gene, so he could manipulate it. And to just summarize, by RNase mapping and RT-PCR, there is a long and a short form. The short form is slightly more abundant. This is the short form. It is exactly 688 nucleotides. The pink region is the ALU element, and this is the polyase signal. It is polydenylated, and he showed that it is largely cytoplasmic, all good things, because that's where SMD resides, it occurs in the cytoplasm. I don't mean good things in the sense that, you know, we are hoping for things that aren't real, but if it's going to function in SMD, it should be cytoplasmic, and it is. Here's the model. In the cytoplasm, there are mRNAs and long non-coding RNAs that confine themselves. We call this long non-coding RNA a half SBS for Stauffen binding site. And that double-stranded region binds Stauffen 1, and we know Stauffen actually stabilizes the duplex. And then when translation terminates normally, upstream of the Stauffen binding site, UPF1 associating with Stauffen triggers SMD. And I'll just show you, I haven't, maybe 5 more minutes?
Yeah.
Oh no, I'm over. I'm over, aren't I?
No. I think you started at 3:30, right?
Okay. I'm almost done. Let's just focus here. So the blue line is Stau siRNA. And while downregulating Stauffen does nothing to the long non-coding RNA abundance, so the long non-coding RNA is not degraded during SMD, these mRNAs, these SMD targets are serpine and FLJ. And they're also, because when Staufen's upregulated, their mRNA abundance goes up. And when the long non-coding RNA is downregulated to about 60% of normal, the SMD target abundance also goes up. So Stauffen and the long non-coding RNA contribute to their decay, and they do so in a way that depends upon the serpine or FLJ 3' UTR. So he localized the regulatory element to the SMD target's 3' UTR. One of the other things Gong did was to actually ask if he could purify a complex that would be predicted if the model is correct, and that is the long non-coding RNA together with the targets, SMD targets that it anneals to, Stauffen and UPF1. And the answer is he can. The way he did this is he inserted 12 copies of the viral MS2 protein sequence into the long non-coding RNA and co-expressed that with a flag-tagged protein that had the MS2 coprotein as part of it. And he did an immunoprecipitation using the anti-flag, was able to purify the whole thing. And let's just focus on the anti-flag IP, where he purified the flag-tagged protein and the long non-coding RNA, and together with it, Stauffen and UPF1, and the 2 mRNAs that he predicted would anneal to the long non-coding RNA, but not irrelevant mRNAs. And IL-7R is another SMD target. It just doesn't have an ALU element with complementarity to that particular long non-coding RNA's ALU element. So we call this long non-coding RNA a half SBS RNA. We call it half SBS RNA1. And this shows, by doing that same purification, that when Stauffen is downregulated, there's much less of the SMD target brought down, so that Stauffen stabilizes the duplex. And then there's many long non-coding RNAs that through their ALU element can bind to the ALU element in an SMD 3' UTR and trigger SMD. I just showed you an example of half SBS RNA1 that can regulate at least 2 different SMD targets. And here's an example of a single mRNA whose ALU element can be regulated by 2 different long non-coding RNAs. So it's a very complicated network. And we're very much interested in understanding how these long non-coding RNAs are regulated. And then finally, just because an mRNA has an ALU element in its 3' UTR that has a very good complementarity to a half SBS that we know regulates the SMD, triggers the SMD of another mRNA, doesn't mean that mRNA is also an SMD target. And the same can be said for microRNAs and their targets. So the bottom line is BAG5 mRNA should anneal to this long non-coding RNA, and it's not because BAG5 mRNA doesn't make it to the cytoplasm. It doesn't co-purify with Stauffen. It is translated, and I'm happy to take questions.
Thank you.